All right, thanks to Nevin and Trey for inviting me here, and I've really learned a lot. This has been really fun today. So I'm going to tell you about something a little bit different today, and I'm going to tell you a story of work that we've been doing in yeast for the last several years to try to understand something about what happens when genes are overexpressed. And so to start, I have to give my conflict of interest. My lab is not funded by NCI, but we study cancer using yeast anyway. By the way, it's in vivo. So this is my second obligatory joke slide. And Carol Privis is one of my colleagues at Columbia. And there was a paper in Science, I think, a couple of years ago. Elephants have 20 copies of P53. And they don't get cancer, okay? So that's interesting. Yeast has no copies of P53. Yeast is cancer, okay? So that's my, that's why you should think about what I'm going to tell you about. So what we're going to do is we're going to try to understand, and this all goes back to synthetic lethality and Hartwell and Friend, and I'll show you that in a minute. But one of the things that we're interested in is what happens when genes are overexpressed. Now, all of you know that we think about knockdowns as being really important, but actually overexpression has just as much of a consequence sometimes as knockdown. And so I'm going to tell you about the fact that genes are conserved from yeast to humans helps. And what we want to do is we want to figure out the pathways that are disrupted when genes are overexpressed, because those can lead to new biological and eventually drug targets. And we can do this in yeast, and it's rapid and cheap, by the way. I should put that in bold. And many of these interactions, as I said, are also conserved. So it goes back to synthetic lethality, and just for simplicity, we'll draw it as two pathways, the black and the blue pathway, and you all know this. If the black pathway is knocked out and the blue one works, the cells are fine, and vice versa. But when you knock out both, cells die. And it was Hartwell and Friend who said maybe this could be useful because cancer cells often have the blue pathway knocked out. And so if you can figure out what problem in the black pathway might be druggable, then you can create a drug. It won't bother the normal cells, but the cancer cells will die. And the poster child for this is BRCA1 and BRCA2 mutants knock out the blue pathway, and PARP inhibitors knock out the black pathway. And so what I want to tell you is, and this goes back to some work that was done by Phil Heater and Joachim Lee about 20 years ago, and they actually showed that synthetic dosage lethality is something that you can also observe. And so the idea is, again, if you knock out a gene in pathway, on the black pathway, you're fine, and if you overexpress a gene in the blue pathway, it may actually disrupt the blue pathway. And so what we can do is we can look for synthetic interactions where overexpression of one gene is synthetically sick when you knock out another gene. And so that's what we're going to do, and that's what I'm going to tell you a little bit about today and how we did some of that work. So the notion is we can screen a set of mutants, and of course we do it in yeast because there are 4,800 non-essential genes in yeast, and we actually can throw in some of the essential genes as well as if we use temperature-sensitive mutants at a semi-permissive temperature. And then what we're going to do is we're going to discover the black pathways for these genes that are overexpressed. So that's the point. And so the idea, of course, at the end of the day is those will become interesting drug targets. Okay, so this slide's a little busy, but the idea is find genes that are overexpressed, and what we've used, and I'm not going to go through all the details, some of this stuff has been published, but what we do is we look at data for whether the gene has been amplified and also whether the mRNA levels are up. And so that's our criteria for whether a gene is overexpressed. The real gold standard is to have protein data. We have that for many things, but we don't have that all the time. The gene doesn't have to be a driver, by the way, in this case at all. But we like sometimes for there to be evidence for complementation. And if you kind of look on this list, one of the genes right at the top is CKS1, and CKS1 actually was discovered in yeast. The homologue in humans is very similar. I'll show you that in a minute. But the interesting point is that CKS1 is overexpressed, for example, in liver cancer almost 20% of the time. So again, when I talk about this, this is our precision medicine kind of idea, because we're going to try to figure out how to deal with patients that might have particular genes overexpressed, et cetera. But just why is CKS1 an interesting gene? This is the homology between CKS1 from Saccharomyces, Pombe, humans, C. elegans, Drosophila, et cetera, and you can see it's highly conserved. And what this gene does is it's part of the CDK that actually attracts phosphorylated proteins to the CDK, singly phosphorylated, so that they can get multiply phosphorylated. So another gene that I'm going to tell you about is RAS, okay? And RAS doesn't really need to be overexpressed, but by using our system, we can introduce activated RAS into yeast. And the reason we picked RAS is, of course, as you can see here, RAS is overexpressed in lots of different cancer cells. And the other reason that RAS is interesting is actually shown on this slide. So yeast, it turns out, has two copies of RAS, RAS1 and RAS2. And if you knock them both out, the cells die. But interestingly, if you substitute the human RAS into the double knockout, the yeast cells are perfectly fine and survive. Similarly, the activating RAS mutation that's been discovered is here. In yeast, it would be VAL19. In humans, it's VAL12. If we make the VAL19 mutation in yeast and we put that into mouse fibroblasts, it can actually transform the fibroblasts. Not we, but people who have done those experiments. Okay, and the third gene I'm going to tell you about is RAD5, HLTF. The HLTF is the name of the gene in human cells, and it has, I'm not going to be able to tell you much about domain structure, but it has a ring finger domain, a SNF2 helicase, which is actually part of its translocase activity, and HiRAN. The HiRAN domain is actually the homology region between these two genes, and that's what it's named for. And so what does this protein normally do? So here's cisplatin stuck on DNA, and DNA replication is occurring, and what happens is pCNA stops, not pCNA, but replication stops, and pCNA gets modified by RAD6 and RAD18. That's the yeast genes. There are human homologs, of course, that do the same thing. Monoubiquitolates pCNA, and that monoubiquitylation allows translesion polymerases to come in and try to bypass this problem. But if the problem can't get bypassed, then RAD5 or HLTF-like proteins come in. They're part of an E2, E3 complex that polyubiquitolates pCNA, and that allows sister chromatid repair, et cetera. Okay. So the reason we got interested in this, and it was partly from a conversation with Carlene Simprich, is that we realized that in squamous cell carcinomas, HLTF is overexpressed quite a bit. And again, it doesn't have to be the driver. Remember, that's the part of the important part here. So what do we do? So what we do is we take the gene of interest, in this case I'm showing you RAD5, we place it on a promoter that's regulatable, and we overexpress it in yeast. And what we're going to do is we're going to introduce that construct into the yeast gene disruption library. And of course, if we do it by transformation, the guys that die are what we're looking for, but the guys that don't transform give the same phenotype. So we actually had to come up with a very efficient way of introducing it. I'm not, again, this is published, so I'm not going to go through the details. But what we did is we created a yeast strain that we could use as a universal donor. So here's the plasmid of interest. The black chromosomes are all destabilizable. So when we mate it to the recipient gene disruption strain, we make a diploid, but of course it carries a wild type copy of the gene disruption. So we need to get rid of all the black chromosomes. And so all of them have promoters that go through the centromeres, and that destabilizes them, a la Carrie Bloom, again, worked from about 20 years ago. So at the end of the day, after a few days of selection and growth, we can get a yeast strain that has the plasmid and the gene disruption, and we can ask, did you grow or didn't you grow? And so, for example, in the empty vector, everything here is done in quadruplicate because yeast wants to grow, so we want to make sure that we don't get false negatives. And so everything's done 4 times, and what you can see is this strain grows fine when you introduce the empty vector to it, but it dies when you introduce the gene of interest overexpressed. And that's what we're looking for, synthetic dosage lethality, SDL. It's fast, it's cheap, and it can be done in 6 days. And we've also developed a bunch of tools that we can use to look at the colony sizes and actually make some cutoffs and figure out where, whether we've got hits or not. And I'm going to just not tell you much about that, but when we were doing these kinds of experiments, like you all know, what we found is we found this nice tail, and the question, and it's kind of come up today, but where do you cut off this tail? So statistically, we can just take 5% FDR and say, let's cut it off there. But what we noticed is often when you did a screen, all of a sudden you'd look just on the other side of the cutoff, and there's 3 genes that are all part of a complex. And so, of course, what does a geneticist do? You bootstrap them into your analysis. But what we said is maybe that's telling us something. Maybe what's happening is, and this goes back to work that's been talked about here today as well, maybe these guys are all genetically or physically interacting with one another, and that's exactly what we see. Because if we take these 200 hits and look at the interaction map, and we take any random 200 hits from the middle of this, what we see is something that looks like this. And this is true for any set of genes here. We see a huge amount of interactions. And in fact, as soon as those interactions drop off, that's where you cut off your screen, and those are the guys that you want to study. So, if you want to read more about this, we published this paper a few years ago, and you can basically see this right here. So when we did CKS1 overexpression, we got a bunch of interesting things, and I'm not going to ask you, you can look at it for 20 seconds, but I'm going to show you a summary of what we found, okay? So here's CKS1. It's actually important for both mitotic entry and mitotic exit because there's CDK that it interacts with. And here it's involved with regulating the destruction of SWE1, which in mammalian cells is WEE1. I'll show you that picture in a minute. But what we noticed is all the underlined genes are the ones that we found as synthetic dosage lethal when we overexpress CKS1. And some of them are involved in this septum ring formation, which is where SWE1 goes to get polyphosphorylated so that it can get degraded. And that's also regulated by CDC5, which is a polo-like kinase. And what's interesting is, even though these are septum ring genes, they have human homologs as well, by the way. And SWE1 has a human homolog, WEE1, very famous. And CDC5 is PLK1. So our data suggested that if you knock out PLK1 expression at the same time that you overexpress CKS1 in a human cell, it might not be good because it's not good in yeast. And that's precisely what we found. This is looking at apoptosis, so the higher you are, the more you're not there. And here's a strain where we knock down PLK1 with shRNA, and we overexpress CKS1, and we see an effect. I'm supposed to show you that circle here. Again, if you're interested, this is published. But one of the interesting things, and I actually raised this question earlier, what we noticed is if we knock out SWE1, we will completely eliminate the synthetic lethality. So if we get rid of the target of CKS1, then it doesn't matter that PLK1 is there or not. And so it suggests that it's not a good idea to ever combine a therapy where PLK1 would be inhibited and WEE1 would also be inhibited, because those cells are actually going to grow better. Okay, and that's actually shown here. So I just showed you for the summary for CKS1. All of this you can read probably as fast as I can say it. Okay, so let's go to RAS for a second, and I see that my time is running out, so I'll go a little bit faster. But what we found is we found a lot of mutants when we overexpressed, and so when I say overexpressed RAS, it's not so important that it's overexpressed. It's actually because we're introducing an activated RAS, and it's RAS2 that we found in yeast to be most important. RAS1 and RAS2 give overlapping guides, but RAS2 gives the vast number. And again, RAS2 seems to be more important in yeast. So we knew from work in mammalian cells that the ER gets expanded in RAS mutants, and you can kind of see that over here. You see lots of ER in comparing wild type and mutant cells. So that made us think of the fact that RAS2 overexpression might be important. The UPR might be important, and in fact, indeed, that's exactly what we found, because IRE1 and HAC1, which are IRE is the ERN1 target. So here's the unfolded protein response. Unfolded proteins brings together this nuclease, which allows the splicing of this activator, which allows the unfolded protein response genes to all be made. This was all discovered by Peter Walters. So this looked really kind of interesting. And so again, we did, we started to do some experiments. We found nothing interesting until we knocked out MEK, okay, MEK. And so if we knock out MEK, then we recapitulate the same synthetic lethality that I just showed you. So why is this interesting? It's interesting because yeast doesn't have the MEK RAS RAF pathway. So yeast is naturally a deletion for that. So yeast also told us that this is a right way to think about this, which is kind of interesting. So if you look at this is complicated, but what we found is that if you knock out this pathway and you get rid of this guy, you can kill cells where RAS is activated. But when I say knock out, we actually had to remove ERN1. It's not the activity of ERN1 per se, so we actually think it's all part of this pathway right here. All right, so the summary of this part is, again, all of this stuff is conserved. It just turns out that yeast is deleted for this pathway. And so ERN1 looks like it's a scaffold for those, for TAC and TRAF, and it's not the enzymatic activity that's important. So it says, look at June and Junk maybe for interesting mutants here. All right, and I don't have time to go through the last part, but when we did RAD5 overexpression, this is kind of looking at just the complete genetic landscape in yeast. We said, what mutants did we find? We found mutants that just were involved in replication and repair, and a bunch of them I outlined here. I won't go through the details. But again, remember I told you about fork reversal, et cetera. So the way fork reversal gets reformed is by homology-directed repair and crossover resolution, and we found all of those mutants. So we know that's part of a problem, and if we overexpress RAD5, so we see increased genetic recombination. So what does it do for DNA replication? So what we said is, what happens if we inhibit cells with cisplatin? And what you find, so if you knock out RAD5, it's actually very important for getting rid of cisplatin damage. Cells are extremely sensitive. This is increasing dose, and this is fitness. And if we overexpress RAD5, we see exactly the same thing. So this might be telling us that cisplatin might be an interesting drug to be used when cells have RAD5 overexpressed. Okay, and again, this is just a summary of what I told you, some of which I didn't tell you because I didn't have time. So can we use what we just found about yeast RAD5 for thinking about HLTF cancers? I just mentioned it, and we're actually working with Anand in Singapore to look for that correlation. And so I'll stop there, thank the people in my lab. I actually, Dan, I don't know if you're here, but we did get a little bit of money to start this project from NCI. And thank you very much.
Hi, Rod. Great talk. So you were showing your overexpression combined with deletion was correlated with the corresponding proteins being physically associated. What about the other side of the spectrum in terms of knockout, then overexpression results in suppression? Is there any interesting biology on the other end of the curve?
Yeah, so it's a nice question where we don't find too much. We don't find too many situations where we find suppressors of overexpression. And it's interesting that we haven't found very many of those, but we find a lot more hits at the other end of the curve. Yeah, of course.
This is one question I've always wondered about. I don't know if you or anyone else has looked at, is there correlation between genetic interactions between double deletions and a deletion and overexpression?
Oh,
You know, what are,
yeah, no, this is wonderful.
or anti-correlation?
Yeah, so, okay. Unfortunately, I don't have the slide with me. So we did it for RAD5 deletion and RAD5 overexpression, and you find a huge number of common genes. But what's really interesting is the guys that are the outliers. So for overexpression, all the holiday junction cleaving enzymes are out there, but not when you knock down. And RAD5 deletion doesn't cause any effect on genetic recombination. So you definitely see differences, and those differences are important. Yeah, great question. One more question in the back.
Hi, great talk. I was wondering, how generalizable across cancers do you think some of these observations that you're making, like the WEE1, PLK1 story? So there have been studies where combination of WEE1 and PLK1, like in squamous cell lung cancer, has been beneficial to the effect of the drug.
Yeah, so I'm interested to know about that because, and also you have to be careful because PLK inhibitors don't always hit the same PLK. And so it might be one that's not, it might be a polo-like kinase that's not involved in mitotic exit or entry. So remember, our whole picture here is that it was the left side of that picture that was important. So it depends on what polo-like kinase, but that's a really good point. Thanks.